Neuromuscular junction, a functional connection between the distal end of a nerve fiber and the middle of a muscle fiber. The neuromuscular junction, or motor end plate, is what we call the synapse when the target cell is a muscle fiber. The sarcolemma is indented and compared to a handprint in clay. The nerve fiber on each synapse has a bulb-like swelling called synaptic knob, which is the dilated tip of a nerve fiber which contains synaptic vesicles. This knob is separated from muscle fiber by the synaptic cleft, which is a gap of about 60 to 100 nanometers between the synaptic knob and the sarcolemma. The entire junction is enveloped by a Schwann cell, and it keeps the surrounding tissue fluid away. In the synaptic knob are organelles called synaptic vesicles, which are a secretory vesicle in the synaptic knob which contains acetylcholine. In the synaptic knob, these vesicles are also known as a neurotransmitter. Nerve impulses that travel down a nerve fiber cannot cross the synaptic cleft. Instead, it causes exocytosis in the synaptic vesicles, releasing ACH into the cleft. The muscle fibers respond to ACH through the ACH receptors, which are put into the plasma membrane across the synaptic knob. Junctional folds, which are invaginations of the sarcolemma where ACH receptors are especially concentrated, are enfoldings that increase the surface area of ACH-sensitive membranes. Muscle fiber and Schwann cells of neuromuscular junction are surrounded by a basal lamina, and it keeps them away from surrounding connective tissue which passes through the synaptic cleft and fills it. ACHE, an enzyme that breaks down ACH after it has stimulated the muscle cell, which is why we must turn muscle contraction off and relax the muscle.